time. People are still coming in, but I'll just go ahead and start. Hello, everyone. Welcome to the University of Washington Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. This session is currently being recorded, and it will be posted later on our website. There will be a Q&A session at the end, so please add your questions to the Q&A feature below and not in the chat box so that it's easier to moderate. And now I will let Dr. Jarvis introduce our speaker for today.
Good morning, everybody. I've said many times that one of the special pleasures you get in academic medicine is introducing your friends and colleagues to your other friends and colleagues. And in this case, it's a real pleasure to introduce my friend and collaborator, Charles Danko. Charles and I have been working together for about 5 years now, almost 6 years now, on problems related to transcriptional regulation because we think this is a really critical piece of understanding genetic risk for autoimmune diseases. Charles is from the DC area and he got his bachelor's degree in biomedical engineering from Hopkins and then went on from there to do his PhD work at upstate University in Syracuse, New York. I guess he wanted more snow than most of us usually want. Um, and then when he finished his doctoral work in Syracuse, he went over to Cornell to get more snow, but also to work on both wet lab genomics and computational biology. And that's when I first met Charles at a Cold Spring Harbor meeting when he was just beginning his work on using uh, precision run-on sequencing data to not only identify regulatory elements in the genome, but also to begin to understand the language that regulates these elements. And a few years, maybe 6 months before we started working together, I had an NIH grant review that where we had proposed to use chip sequencing to identify regulatory elements. And one of my reviewers, not Charles, he still said, well, they probably should be used global run-on sequencing to identify regulatory elements. So what that tells me, Charles, is somebody actually read your paper. And the rest, as they say, is history because we've had a productive collaboration. We have 4 manuscripts in preparation right now, and I'm really excited to have Charles tell you about his really important work. Charles, you're on.
Well, thank you for that great introduction, Jim, and thank you for the invitation to be here today, and thanks to everyone for joining. Um, so the image in the title slide, I think many are maybe curious about. This is what you get if you ask an AI image generator for a Rosetta Stone for regulatory DNA. It was made by Anita, who's a graduate student in my group, whose work I'll talk about some today. And I love this image because it so accurately captures so much of what we seek to achieve in our lab. So today I'll tell you a lot about mainly technology development projects in the lab. I've sort of centered the talk around that. I will say that a lot of the tools that we develop have implications for understanding basic transcription. And there's a lot of ways that our work interfaces with that too. So if you have questions about that, please do ask at the end. So I'll show a couple today, but not as much as our transcription focused talk. So the way that I motivate all of this work is to understand the central dogma of molecular biology, this basic feature by which DNA is converted into mRNA by transcription and then ultimately into protein by translation. What we study in our group is how DNA encodes the amount of mRNA. And we think that this is an important problem to understand because this is really a key regulator of complex behaviors of cells and ultimately of phenotypes, both within different species or between species or changes in cellular behavior that give rise to a disease, like this mast cell tumor, in this case, in a yellow lab. So gene regulation is encoded by regions that we call transcriptional regulatory elements, or cis regulatory elements. And these occur when the activity, a transcription factor protein, recognizes its DNA sequence. So it recognizes a specific DNA sequence in DNA. So here the cartoon, the gray represents DNA and the ovals represent nucleosomes. And this green shape here represents a transcription factor, which recognizes and binds to its specific DNA sequence motif. When it's bound, it recruits a variety of different coactivators that have a large impact on the surrounding chromatin environment. And they have a variety of tasks, including decorating core histones surrounding the transcription factor with post-translational modifications, as well as recruiting the RNA polymerase II complex and initiating transcription. And you can see, if you look at the distribution of RNA polymerase across the genome, you can see these major stages of gene regulation, where RNA polymerase is first recruited by a transcription factor to a regulatory region, a cis-regulatory element. It's initiated on DNA. That initiated RNA polymerase, initially associated with the pre-initiation complex, begins transcription and transcribes about 20 to 60 nucleotides where it stops, and is a remarkably stable structure known as a promoter proximal pause. Other transcription factors can act to release this pause into productive elongation. And then if this RNA is destined to be stable, through DNA sequence codes that we know through the work of Phil Sharp and others, this RNA polymerase will transcribe for up to a megabase in a mammalian genome, and produce this stable RNA behind it. But it's important to note at this juncture that enhancer sequences which regulate the rate of production of RNA from a distal promoter also undergo this process of transcription initiation and generate very stable RNAs as a result, called enhancer RNAs. So these steps of transcription are clearly visible if you use a genomic technique to assay the position of RNA polymerase in a population of cells at steady state. So here this figure represents the amount of RNA polymerase on the plus strand in red or the minus strand in blue. The x-axis here is position along the genome. So for each position along the genome, the height of the track represents just the amount of RNA polymerase. And you can see for a typical gene near the transcription start site, there's this excess RNA polymerase, which represents the paused RNA polymerase. And then the RNA polymerase, which is transcribing through the gene body here. And then you get termination of that RNA polymerase. You can also see that there's this RNA polymerase that transcribes a very short transcription unit upstream and antisense to a protein coding gene. And you observe this same divergent transcription pattern, not just at promoters, but also at distal enhancers. So as I mentioned, RNA polymerase is transcribing very unstable RNA species that are really very strong markers of enhancers. And this is what Jim's reviewers were referring to, which definitely was not me. So these enhancers can be intergenic, they can be intragenic, and you can pick out these enhancers as well as lots of other different varieties of regulatory function from this type of genomic assay called PRO-seq. And so we think that this has lots of applications. We think that this is a great assay which maximizes the information that you gain from a particular sample. Just to illustrate that point a bit more, these sorts of features that I described in the last slide that are evident from PRO-seq data, you can get to them in other ways too. For instance, ENCODE Consortium has spent a huge amount of effort categorizing a variety of different chromatin post-translational modifications and their genomic distribution across large numbers of cell types and tissues. And these are incredible resources for the community. And to a large extent, they show a lot of these same types of functional units as you would get by looking at transcription. In addition to that, transcription also gives you a bona fide measure of how rapidly you're transcribing mRNA, and therefore it's quite highly correlated with RNA-seq data. It's really a measure of the rate of production of new transcripts. One way that I like to think about this is that RNA polymerase sort of is situated right here in the central dogma between the chromatin level and the mRNA level, and it contains features of both of these different regulatory stages that with the right computational methods, we could sort of suss out. And this makes it uniquely informative marker of gene regulation and genome function across a new cell type that you might want to assay. So I'll talk today about two major vignettes that are actively going on in my lab. First, we'll talk about methods that we have developed to sort of suss out what the biological function of each region of the genome is using transcription as the single marker and applications of these various approaches. And then second, I'll launch into new work that the lab is doing and actively growing on, which happens to be a collaboration with Dr. Jarvis, on interpreting how DNA sequence is influencing the transcription cycle and using these to understand how autoimmune risk alleles impact gene regulation more broadly. So I'll get to both of these today. So first of all, the ideal genomic approach is going to be one that maximizes the information gained using a single experimental assay. That's sort of the basis with which we're coming from a lot of this work. And so we'd like ideally something which measures gene expression, regulatory activity, or epigenomic measurements, so things like histone post-translational modifications or surrogates that represent a similar underlying biological process. And we'd like to be able to gain single cell resolution on all of these approaches. So I'll talk about all of this today, mainly focusing on these last two, which I think are the harder problems. And what our lab is attempting to do with, I think, some success at this point, is to develop machine learning methods for predicting, this is the second part, where we're developing machine learning methods to predict patterns of transcription from primary DNA sequence. So we'll get to that later. But first, this first part. So our goal, essentially, we can think about this as taking these maps of where RNA polymerase is located along the genome and decomposing them into these ChIP-seq-like tracks, which represent the abundance of different histone modifications along the genome. And the assertion is that with a single track of RNA polymerase, maybe we can estimate lots of different chromatin marks that would have been measured separately in independent experiments by ENCODE and other groups. This turns out to be a difficult problem in computer science, as shown by the XKCD. I won't go through it. But has been made easier, especially in more recent years as machine learning approaches have been developed and have really gained some traction and some success in the domain of image recognition, which draws a lot of parallels to recognizing these patterns in the distribution of RNA polymerase. So the first method that we developed now many years ago when I was making the transition to an independent faculty position was called DREG, which stands for the detection of regulatory elements with GRO-seq data. So what we do is we take this input pro-seq data set, and we have a data set from which we know the position of regulatory elements, cis regulatory elements, just generally, including promoters and enhancers. And we trained a classifier to recognize this shape of transcription that's associated with these. We use a discriminative model called a support vector regression to achieve this task. And that regression produces a score as a function of genomic position. So here, it produces scores that are higher within these regions that are also marked by chrome-HMM, which is an encode metric that synthesizes lots of different ChIP-seq data sets. 38 actually ChIP-seq data sets in this particular chrome-HMM track. And DREG represents the location based on ProSeq data of active regulatory regions. And you can see that the two correspond fairly well to each other at this particular locus in IL6R. You can also use the same type of approaches to estimate where transcription, or which transcription factor binding sites are going to be occupied. So the slide shows the occurrences of 4 DNA sequence motifs recognized by the transcription factor ELF1 in a region near the KIF14 gene. And you might predict that this motif here, which is located in a region that is actively serving as the promoter of KIF14, is most likely to be bound than these other 3 here, because it's in this active regulatory region. And indeed, when you do the ChIP-seq experiment, that's exactly what you see. So Lauren Cho, a former graduate student in the lab, used very similar computational methods that we introduced in DREG to show that you can use this type of divergent transcription signature to get really good state-of-the-art at the time estimates of what fraction of those motifs are actively bound by that transcription factor versus unbound. So this gets not only at which regions are cis-regulatory elements, but what transcription factors those regulatory regions are binding. And then finally, a couple years later, Zhong Wang, who was a programmer in my lab for a long time, introduced variants of these approaches that estimate histone modifications directly. So the input to this support vector regression is the pattern of transcription within a locus. And then the support vector regression is trained to estimate the abundance of histone modifications every 10 base pairs along the reference genome. And we trained models for 10 different histone modifications, which we divided into different classes. So those enriched near cis-regulatory elements, those which are indicative of gene bodies, and repressive marks. And I'll say right now at the outset that the only one of these that didn't work at all was H3K9 trimethylation. So this slide shows the correspondence between the experimental and the estimated H3K27 acetylation, which for those who don't know is a marker of active promoters and active enhancers that's widely used to sort of categorize enhancers as active. And it was the imputation is based on this data representing transcription in about a 2 megabase region on a holdout chromosome. You can see that the experimental data and imputed data are fairly similar to one another. And this is true for lots of different histone modifications, including histone modifications that aren't very broadly analyzed, like this globular domain modification H3K122 acetyl, which has been proposed by Wendy Bickmore's group to be a better marker of active enhancers than K27 acetylation, as well as very different signatures like H3K4 monomethylation, which is best known as a marker for enhancers, and H3K36 trimethylation, which marks the body of active genes. And to my surprise, we even had good traction for the repressive histone mark H3K27 trimethylation. This is true at a 2 megabase scale. This is also true if you zoom in to sort of a 10 kb region. And in fact, the classifier is able to come up with a lot of the fine scale detail in these types of histone modification tracks, like this depletion in signal in the nucleosome-free region, and the asymmetry between the amount of H3K4 trimethylation in the coding and the upstream antisense strand that is sort of characteristic, in this case, of a promoter. So I was really excited from the start of this project about what we could learn about the function of these histone modifications. And so I spent a lot of time looking in the genome browser for cases where the classifier got it wrong. And one of those types of examples is shown in this slide. So here, this slide, this track shows ProSeq data in a region. This slide shows DNase 1 hypersensitivity by ENCODE, which is a marker of open chromatin. And then the ENCODE ChIP-seq data did not have any evidence whatsoever of H3K27 acetylation at this regulatory region here, which was really exciting because DHIT predicted a lot of H3K27 acetylation at this particular region. So I was all full of experiments where we would ectopically use CRISPR type approaches to recruit H3K27 acetyltransferases to this region and see how it affected transcription and lots of different perturbations that we could make to understand what the role of this histone mark is at this particular locus. But before going down that road, we of course did our own experiment to ask if there was indeed a lack of H3K27 acetylation there. It occurred to us that the alternative model for why we might get a deviation is just differences in cell culture conditions or the hands of the individual who was culturing these particular lines. Even though these are both the same type of cell, there's a lot of environmental influences that can impact both transcription and histone modifications. And if those aren't controlled very precisely, this is the type of pattern that it could give you. And so when we did the experiment in our own hands, we did not sequence this as deeply, but very clearly we see that there's lots of H3K27 acetylation here, better agreeing with our imputation model than with the ENCODE ChIP-seq data. And so this was true everywhere that we found major discrepancies like this. And so the takeaway that we took home is that transcription is really tightly linked with these histone modifications, at least in the cell types that we're looking in. And it reflects probably the same underlying biological factors that influence both of these two processes of transcription and histone modification, with very little discrepancy between them. So the other exciting thing that we can do with this is that once we have these models trained in one cell type, everything I've shown you so far is a K562 cell, which you could argue does not even really reflect a human anymore. We asked whether these same models are accurate in other cell and tissue types, and even in other species where we have data. And indeed, when we do this comparison, we take the K562 model and we use it to estimate histone modifications using transcription from GM12878, HCT116, HeLa, T cells, or even primary liver tissue from horse or embryonic stem cells from mouse. All of the active histone modifications showed about the same accuracy as in the training cell type. And the way that I interpret this is that the relationship between histone modifications and transcription is a deeply conserved biological process. And so we're able to train a model in K562 and then adapt this model to make predictions in any cell or tissue type. And what that lets us do is collect the data in a clinical isolate, for example, and then impute all of the ENCODE data that you could get from that clinical isolate using just a single molecular technique, this run-on technique called ProSeq. Yeah, so that is, I think, really has driven a lot of our excitement into these molecular techniques. And of course, the problem with them is that they require a large proportion of cells. They're hungry. They require millions of cells as input, or at least 1 million cells as input is what we typically use in most cases. But we'd like to be able to make single cell inferences using these techniques. And so a graduate student named Tinyi Chu sort of thought through this problem and had the hypothesis that he could address this using computational methods. And the goal that he set out to achieve, in essence, is to basically extract information about regulatory activity in different single cell populations by integrating CRO-seq or RNA-seq data with a single cell variant that is correlated with those other data types. And so if we think about the advantages and the disadvantages of each one of these approaches, single cell RNA-seq is incredibly informative about individual cell populations, but it has a lot of disadvantages associated with it as well. There's biases associated with dissociation, depending on how you actually do that. They're incredibly expensive and remain so to this day. And then there's relatively low coverage per gene or per peak if you're using single cell chromatin accessibility measurements. They're rife with dropout and zero inflation that we'd like to be able to correct. Conversely, bulk measurements are scalable. They're cheap. There's thousands of existing data sets that we can apply them to. And the only disadvantage is that there's no single cell information. And so the further we have gone on to this project, the more we've converged to an ideal experiment to analyze, say, a new disease system would have the following properties. We'd collect single cell data from a limited number of individuals that sort of represent as much of the variation as you can capture from a cohort, maybe 3 to 10 samples. And then you'd collect a very large number of bulk samples, 100 if you're an individual lab might be reasonable with these bulk approaches, or 1,000 if you're a course consortia, many thousands potentially. You could scale these approaches too. And using these, basically using computational data integration strategies, could we then take the bulk data, use the single cell data that's well matched to it, and extract as much information as possible about each cell type in the bulk data. So Tigny developed a method to do exactly that, which is called Bayes Prism, which was published now several years ago, where essentially he took a single cell RNA-seq data set, and he took a bulk data set, or many bulk data sets, and he developed a Bayesian inference approach that leverages the information in the single cell RNA-seq data to split apart the counts for each gene that you observe in the bulk data set into the posterior counts you expect, conditional on the single cell data, for each of the cell populations that's represented by the single cell data. So in other words, what he's doing is like a prism, he's taking counts from individual genes in a bulk sample and assigning each one of those counts to the cell type most likely to have given rise for that particular count. So this does two things for you. First, it's a really good estimate of the cell type composition in a sample. And second, each of the rows here, which you use to get that cell type composition, is an estimate of gene expression, the posterior estimate of gene expression within that particular sample. So to prove that this worked, Tigny did a ton of benchmarking that's described in his paper. And what I found most compelling was a glioblastoma data set with two independent single cell data sets of the same tumor type that were collected by different labs. And so what Tigny could do is he would know the ground truth of how many cells of each cell type were present in the one data set. And he could use the other data set to accommodate batch effects as best as possible to deconvolve a pseudo-bulked version of the other single cell data set where he's sort of lost the labels of which cell each read is from. And what he showed is that he can with remarkably high accuracy, he can infer the cell type fractions of tumors, of myeloid cells, tumor infiltrating, probably microglia rather than a macrophage, T cells, oligodendrocytes within the pseudo-bulked data set. In addition to that, glioblastoma is a really interesting tumor because the malignant cells from each patient have a very distinct gene expression profile compared to any other patient. So the information that he gets from the single cell data that he uses as a prior is not directly informative about the single cell data that he applies this to. So he asked how well he could estimate gene expression within the malignant cells of each tumor. And this is a UMAP representation which basically show, so each patch here are cells from a different patient. There's about 28 patients in this data set. And what you can see is that the inferred gene expression estimates by Bray's prism tend to be very close to the ground truth that, he's able to estimate. That he knows based on actually having the single cell equivalent in this particular case. So this convinced us that this Bayes prism algorithm does a remarkably good job of estimating both cell type composition and gene expression. So one of the vignettes that Tigny told in his paper is he looked at the effect of myeloid cells, just the composition of myeloid cells across a lot of different tumors for which there were single cell data sets when he started this project. And these include glioblastoma, head and neck cancer, and melanoma. And what he found is that in melanoma, high macrophage content generally had, patients with a higher macrophage composition generally had a better prognosis. And in glioblastoma, we didn't quite get statistical significance, I believe, after correcting for multiple testing. But if anything, there was the opposite relationship. So to address why myeloid cells appear to have different prognostic effects in different tumor types, he broke apart gene expression within the macrophage and asked whether myeloid cells in these different tumors appeared more M1 or M2-like using gene signature matrices that were found in other papers. And what he found is that the difference between M1 and M2 did a remarkably good job of sorting out differences in macrophage polarization within the different cancer types. So glioblastoma tends to be much more M2 and less M1-like, whereas melanoma tends to be much more M1-like and much less M2-like in the gene expression signature within these macrophage populations. And in fact, this macrophage polarization signature was an even better prognostic indicator than just macrophage composition alone. This was true in melanoma, and this was true in the other two skin cancers as well. And so this is, I think, a very powerful demonstration that incorporating these estimates of gene expression from bulk data, but where the gene expression references the estimated expression within just one of the cell types of interest, is an incredibly powerful approach. So a key goal of the lab since then has been attempting to apply these techniques to deconvolve using single cell attack-seq data, estimates of pro-seq gene expression. And so we've, again, using collaborations with Mariano Villapano at SUNY Upstate, we've collected single cell attack-seq data in actually, so the slide is 3 GBMs, but we have 11 now. And the pro-seq data we've collected from matched samples where we have the single cell attack and then the same nuclei have also gone in to create a pro-seq data set. And we can do a remarkably good job of deconvolving pro-seq from the single cell attack-seq data in most circumstances. And then for each one of these different tumors, we can estimate the amount of chromatin accessibility that we have in that particular tumor. And then we've collected bulk CRO-seq data for 70 over, actually now over 100. The slide is the first 70 that we sequenced, different glioblastomas, and we can estimate cell types and chromatin accessibility in each one of these different cell types within this data set. And so I think this is just also a powerful demonstration that individual labs using these techniques and carefully considering which genomic assays they can do can create some incredibly large data sets that really give us the power to drill down on what the mechanisms of a lot of these disease processes actually are. And I'm very excited to see these applied by you guys. And we're happy to help you get started if there's ways that we can. So in summary, in this part of the talk, ProSeq is a single molecular tool that lets you measure gene expression, transcriptional regulatory regions, impute histone modifications, predict transcription factor binding, and then possibly even leveraging these Bayes prism and similar tools to decompose the bulk patterns that we get in ProSeq down into the individual cell types that contribute to them. Yeah, so now I'll switch gears a little bit and talk about measuring the link between DNA sequence and what that means for transcription at each locus. And that's where we introduce our Rosetta Stone for reading regulatory DNA. So the core problem here is that DNA sequence patterns of transcription and chromatin accessibility are controlled by these transcription factor binding motifs, and each one individually isn't very informative about genome function. So this motif here I'm showing you is ELF1. It's recognized by a transcription factor called ELF1. And what you can see in the slide is that the height of each base corresponds to its importance for ELF1 recognition. So the bases here in the center are generally CCGGAAG. But there's a lot of bases out here on the periphery where they can take either a G or an A, or sometimes even a C or a T. And even bases here in the core often mismatch the preferred ELF1 base at that position at true ELF1 bound regions by ChIP-seq. And so as a result, even though this is 12 base pairs of DNA sequence size, there's actually only about 6 base pairs of information contained within the sequence motif. And it's hidden among a genome of roughly 3 billion nucleotides. So depending on where you put your threshold, there could be millions of motifs that match, and most of them never bind ELF1 at all. And so you're sort of left with this needle in the haystack problem where you're not sure which ones are real and which ones are not. And so does DNA sequence even have the information content to encode these regulatory programs at all? So there is a model where it's not just the individual motif that's important for encoding a regulatory program, but the context with which that motif is organized relative to the binding sites of other transcription factors and maybe core promoter elements is really important for driving its regulatory activity. And there's a few classical examples where this is incredibly important. And this being a rheumatology division, many of you are probably familiar with this interferon beta enhanceosome concept, where the order and orientation of a bunch of the DNA sequence motifs within this enhanceosome are critically important for its function as an enhancer. And so this has spurned what we call a motif syntax model, where the organization of the motifs themselves carries additional important information that make the difference between whether it's an active cis-regulatory element or not. And there's some lovely work, in particular by Emma Farley's lab, that has really run with this concept more generally. But the problem with it is that it's relatively rare. And there's a lot of people in our transcription community who believe that this just doesn't happen very often. And it's more the order of the actual, or just the identity of the individual DNA sequence motifs that are important, right? Where it doesn't really matter what order or orientation they're in, but just so long as you have the right combination of motifs binding, that is sufficient to induce transcription. So that's sort of the context on the biological debate that we're working from as we're coming at some of these problems. And so Adam He, who's a graduate student who just defended his thesis this fall in my lab, his approach toward this problem was to train AI models that take as input these maps of transcription and output so they train to take as input maps of transcription and learn patterns of DNA sequence features which appear to underlie those patterns of transcription using more modern and quite sophisticated machine learning models. The data type that he's working from that I probably need to say something about is called ProCap. So most of what I've shown you is ProSeq data, which measures the position of RNA polymerase no matter where that is. ProCap is a variant of ProSeq, which only looks for the 5' end of capped RNAs, and in doing so, it collapsed all of these active polymerases down to the transcription start site that gave rise to it, I think is the way of thinking about it. And the reason that Adam uses this data is because it gives him better resolution to find intergenic enhancers without having signal from the transcribed gene it's inside of can found that. So that's what he's after, is this local influence of gene expression. And so he trained a model called ClipNet. This was really his primary contribution as a graduate student. And what ClipNet does is it takes personal genomes from 58 different individuals, and these personal genomes are matched to ProCap data. And so a single model, ClipNet in this case, learns how patterns of transcription within a 500 base pair window map to the DNA sequence region 1,000 base pairs, where the two are centered in the same place. So I hope that's clear. And the idea is that this ClipNet model will learn the DNA sequence features that allow it to estimate transcription patterns accurately. And then we can interpret those models to pick them apart and figure out which features in that DNA sequence are important to one another. And that's sort of what I'll go through in the next few slides. So ClipNet is a 9-layer convolutional neural network. It's very similar to Chrome-BPNet from Anshul Kundaji's lab. If you're familiar with that, that really inspired a lot of the architectural choices that we made in this model. I think the major differences are that it takes what we call a 2-hot encoded matrix at the bottom, which represents the identity. So each column represents a position in the DNA sequence. And 0, 1, or 2 represents whether that particular individual has 0, 1, or 2 copies of an A, a C, a G, or a T at that position. So this individual here is heterozygous for C at this position, but they're homozygous for CT at this position over here. So I hope that's clear. And then the model, the intuition for the model itself is the lower layers learn DNA sequence motifs, and the upper levels learn combinations of motifs that influence either the organization with single nucleotide resolution of transcription or the quantity of transcription that is output. So the model predictions are remarkably good. So here I'm showing examples where in the 500 base pair window that we predict near the INDES6 promoter, the observed signal from the experiment is here, and right below that is the prediction. And in all of these cases, which are not cherry-picked, these are randomly selected, the model does a remarkably good job of picking out where transcription is located there. In the interest of time, I'll just gloss over some of the other QC metrics that we did. We get a pretty good correlation at the quantity level. Despite the fact that we're only using 1 kb of local sequence, it's sufficient to explain a huge amount of the variance in transcription, which I think is fascinating. And we're also getting close to biological replication in profile, which is sort of where transcription is located, just the correlation between the observed and predictor tracks. So happy to take questions about that. Adam has shown in this separate paper, actually, that training on personal genomes does a much better job of allowing the model to predict what the impact of DNA sequence changes are. So here he's compared for transcription initiation quantitative trait loci, so a single nucleotide change, which appears to influence transcription initiation. He shows that he can predict those much better on a personal genome trained model than a reference genome trained model. So this, I think, is also a really important element of our approach to this particular problem, is this matched genome and functional data comparison. And then the most important part of all of this is that then once the model is trained and we validated that it can do a reasonable job of predicting out of sample initiation patterns, we can ask the model what it thinks is the most important DNA sequence positions that influence the pattern that it gives it. So shown here are what we call Shapley values, or deep Shap scores, which represent how important each nucleotide on the x-axis is for the prediction that is shown here for the IRF7 promoter. So there's two predictions for these sequence features. The top one depicts the estimates associated with the amount of transcription, and the bottom one depicts the estimates of importance for the shape of transcription. So for the bottom one, why does the model think that most of the transcription is located right here at this position? Yeah, and by and large, most of these correspond to the motifs of known transcriptional activators or core promoter motifs. So here we see this is a Tata box, and this one here, note that this is the reverse complement. And this here right under the transcription start site is the initiator. So using tools from Anshul Kundaji's group, we can categorize these into different transcriptional activators and repressors. So this is a lymphoblastoid cell line, sort of a modified B cell, and you can see that ETS is one of the most important transcriptional activators that is responsible for quantity, which makes sense, I think. And IRF is another, and that also makes sense. And so largely we get what you expect. These motifs we do find have a strong positional dependence with one another. So the way that Adam approached this is he would delete 10 base pair regions by shuffling the sequence of that 10 base pairs and observe the effect of that 10 base pair sequence disruption between the wild type and the mutant predictions. And what he found is that there's roughly 5 different positions that are really important and play differential roles in either the profile on the top or the quantity of transcription on the bottom. And these 5 different regulatory regions are comprised of 3 which govern the pre-initiation complex, these 3 here in the center, as well as transcriptional activators, which are typically found or peaking at minus 50 base pairs relative to the transcription start site. So Adam has asked if there are ways that the organization and abundance, sort of what are the syntax relationships between these core promoter motifs and the transcriptional activators, using a technique called deep feature interaction maps. And what this amounts to is making a computational point mutation and evaluating what the effect of that point mutation at a source nucleotide is on surrounding nucleotides around it. Where the mutation of the C has a large impact on the A's and the TA around here. And so therefore we say that this is potentially a candidate for a nonlinear or epistatic effect between these two sequences. And then we display these as a heat map. So here's the heat map for IRF4, I believe, protein coding promoter. And you can see that this initiator here has strong evidence of interactions with a Tata box located right here, as well as with these SPKLF motifs located further upstream. So this provides very clear evidence for interaction both between the core promoter motifs themselves, as we might have expected, but then also with the transcriptional activators of SPKLF. So sort of supporting that hypothesis that there is a syntax relationship, particularly between different transcriptional activators. I'll gloss over this in the interest of time, but the other point that I want to make is that different transcriptional activators seem to have very different positional profiles relative to that transcription start site. So some show relative positional independence. In other words, they're high across relatively large 100, 150 base pair region within that nucleosome-free region, whereas others are quite peaked. CREB, TFEC, SPKLF, and YY1 have quite precise positional dependencies, which are picked up by the model. And so one hypothesis that I think is really neat is that perhaps these transcriptional activators have a positional dependence which is important for conveying the function of that transcription factor on its role in influencing the transcription cycle. Finally, there's lots of applications of these models in interpreting regulatory changes that are a regulatory change in sequence that has a regulatory impact and ultimately has a phenotypic consequence. So could these syntax relationships more broadly, or more broadly, the transcription factors that underlie them have phenotypic consequences, basically is the question. And so this is a project that we're working with Jim Jarvis on. We're focusing on CD4 positive T cells, which are very highly enriched for a lot of autoimmune and allergic disease genome-wide association study variants. And with Jim, we've collected DNA sequence from 6 individuals, as well as ProCap, ProSeq, and MicroSeq data. And then we also have ATAC-seq data, which represents chromatin accessibility in a different, from a different cohort, which was already generated by Aviv Regev's group that we're incorporating as well. And just so just to give you an example, here near the PTPN2 gene, this enhancer contains a juvenile idiopathic arthritis risk allele, this one right here, that we'd like to be able to categorize as having an effect on one of these stages of gene regulation. And so the way that we're approaching this problem is we're training 3 different machine learning models in this cell type. We're using models of chromatin accessibility, so Chrome BPNet on the ATAC-seq data. We have models of chromatin accessibility based on ProCap data, so ClipNet, similar to the B-cell model I showed you in the last few slides. And then we have a ProSeq model where we're predicting the effect of each variant on the position at which RNA polymerase II pauses, which we're using as ProSeq. And these 3 different regulatory steps represent different stages on which a transcription factor can interact with the transcription cycle to influence gene expression. And so we find lots of DNA sequence variants that influence chromatin accessibility. And here's one example in the slide. So here, this RS6776243 is a risk allele for celiac disease. And this SNP impacts an NF-kappa B binding site right here, where this T disrupts the NF-kappa B binding site, has a major effect on NF-kappa B binding, and is predicted to have a huge effect on chromatin accessibility. And then in a collaboration with Jim's group, we see that it also has an impact on a massively parallel reporter activity, which is more direct measure of enhancer activity. So that's really exciting. But a lot of the examples that we're finding don't just hit transcriptional activators that we would conventionally think are important for this process. Here's an example which is a risk allele for Crohn's disease, where the SNP in question disrupts a downstream promoter element, DPE, part of the DNA sequence which influences the binding, or thought to influence the binding of TF2D in the pre-initiation complex. And this is located in an enhancer region far away from any promoters. Here's another example in juvenile idiopathic arthritis, where there's a new initiator that's created by this T, the introduction of a T nucleotide here instead of a C. So there's lots of cases where the initiation signal for an enhancer RNA seems to influence autoimmunity as its mechanism of action, which I think is an incredibly interesting result. And then we have at least one case where there is a pausing motif here for inflammatory bowel disease. This SNP here influences this motif, and by doing so, the amount of RNA polymerase which is paused at an enhancer element. And this also was turned up by the MPRA. So I'll stop there. In conclusion, I showed you today that chromatin marks can be imputed by RNA polymerase. There is a complex syntax between transcription factors and the core promoter. eRNA initiation and pausing may have an influence on enhancer function. And I've shown you models that we think serve as a Rosetta Stone, allowing us to decode regulatory sequence and predict how different risk alleles for phenotype associated genetic variation influence the process of transcription. So thanks to my group. This was last year. This is Adam, Anita, work on a bunch of that work that I talked about today. Ed collects most of the ProSeq data in the lab. We do Halloween right in our lab. So thanks, and thanks to all of you for listening.
Thanks, Charles. That was absolutely fabulous, and I have more work for you to do based on
Uh-oh, sounds like fun.
based on this Bayes prism idea. Um, Danielle, where do I pick up the Q&A here? Let's see. No open questions right now. Anybody want to dive in?
Yeah, I don't see any questions in the chat, so people can start typing, or if they want to speak, we can allow them to speak too.
I'm gonna suggest a thing. I hope that my colleague Sayori Sakaway is listening. We're very interested in the genetic and epigenetic mechanisms that determine treatment response in juvenile arthritis. I know that it's shocking that we don't actually know why our drugs work. And Sayori and I were in the midst of writing a grant to use single cell sequencing to answer that. But at $7,800 per sample, it's very hard to do that and get all the information. So I was thinking that maybe the Bayes Prism approach would work for that. So my question is that could you also integrate one more layer of data, say our CD4 positive T cell data on top of that? So you have single cell sequencing data, CD4 positive T cell bulk data, and then peripheral blood mononuclear cells. Could the model handle that as well?
Yeah, so you're suggesting sort of a separate population of bulk CD4 positive T cells and PBMCs and
Yes.
trying to use a single cell atlas to sort of decompose both. And yeah, you can do that for sure.
Okay.
Um,
All
yeah,
right.
there's a variety of tips and tricks that we've learned since TNE published that sort of how to handle cells that are missing from the bulk, from the reference, and vice versa. So
Yeah. Well,
yeah,
that's why I thought the CD4 positive T cell da- data might also add better train the model. And also, if you only have to study 10 samples, say 15 samples, you could count 15,000 cells to get more information, right?
you mean for
Or
the single cell atlas that you put into it?
yeah.
Yes, you could. The way that these assays are usually applied is to collect a large number of cells with relatively low quality data at a time. And so you usually don't exert as much control over the number of cells that you sequence as you used to with older approaches like SmartSeq, if that makes
Okay,
sense.
we can talk good. Dr. Mustelin asked whether you've looked at the transcriptional start sites and regulation of endogenous retroviruses.
Yeah, that's a great question. We have so endogenous retroviruses have been studied more extensively by my colleague Cedric Fachat with these same techniques. And yeah, you can see cases where the endogenous retrovirus acts as a promoter and transcribes a copy of itself. They also lose their, I believe it's I'm not a ERV expert, but you lose a portion of the ERV some proportion of the time by recombining it out between the two sort of flanking regions. Those you can also see. The trick with all of those is that you have better power for more ancestrally and older ERV insertions because they've had time to accumulate mutations that you can use to map them uniquely. So depending on what his application is, and I'm glad to talk to him about it,
Yeah.
that could be a consideration, is unique mapping to the
And
PRV itself.
he also asked, do you have access to data from neutrophils? And I,
Ah,
we're going to get that.
we'll get it.
We do have neutrophil differentiated HL60 cells that we're just in the middle of doing, but we are going to eventually get neutrophils. And then Rayan Najjar, with whom you're going to be speaking shortly, asks whether you have the question is, have you explored RNA polymerase 3? And
Ah,
do the assay by positioning RNA polymerase 3.
that's a very insightful question. Yeah, so ProSeq picks up all three RNA polymerases, and or five RNA polymerases in plants. It works by incorporating a labeled nucleotide through the actions of that RNA polymerase and then fishing out the RNA. So we have no specificity for just a single polymerase. And yeah, we see lots of great signal near Pol3 transcription units. Yeah, for sure. We can ask lots of questions about how those work. It's less my specialty, but there's lots of people using the technique for that particular application.
Yeah, and we hit any of our data sites, data sets that we've generated, ProSeq, ProCap data, Rayon, you're welcome to get a look at. Other questions? All right. Well, Charles, this has been a lot of fun, as usual. I left you an email about the Bayes prism, and we need to talk about that more.
That's
And
great. Yeah.
I think this is a brand new way of looking at the genetics of autoimmunity. I think the field really has stalled since the completion of GWAS, and that this approach of looking at getting right down into the nitty-gritty of how variants on haplotypes affect transcription and the different stages of transcription is going to be really uh, a big change for the field, and a huge contribution to how we understand how autoimmune diseases develop. So thank you for being here with us. It's been really fun. And as I say, I hope I'll talk to you tomorrow. I left you
That
an
sounds
email.
great. Yeah, sounds great.
All
Great
right.
to